We normally do not have talks like we do whatever it takes to get this done. It was originally advertised for originally advertised for 3 o'clock, but due to circumstances that we changed to this time. So thank you very much for making it. We have in our next 2 persons today, we have Dr. Dougbeh Nyan. He is an American librarian, he's an inventor, and for what we do here, that is something that we need to get into, okay, inventions. So it's a privilege to have him here to tell us about some of the work he's doing in diagnostics. And he's here with engineer Akofa Batlange. She's been in practice for the last 17 years and actually does a lot of consultancy work. So they are here together, but he's going to do the presentation. So I would hand the microphone over to Dr. Nyan to take us through what he has for us today. Doctor, you're welcome.
Thank you very much. Good afternoon to the director of the Institute, fellow scientists here present, and the very astute colleague who made this arrangement, Dr. Akofa Batlange, and to you, fellow students. I say fellow students because we are all still in the learning process, no matter how professorial we become. When you look at the COVID-19 statistics, it will show that Africa has been doing well for whatever reason. And so most of the COVID-19 regulations seems to be far more relaxed on the African continent than other places, particularly the United States and Britain, where Omicron BA.5 variant is still raging. And so I see a few of you with masks. I will ask you also then to permit me to bring this mask down so that I can speak to you much better and you can see my face. So, I will be speaking with you today on the challenges and prospects of in public health response to outbreaks in West Africa. In Africa, the West is in parentheses to sort of emphasize our region. The case of Ebola, COVID-19, and Monkeypox. There are many more infectious diseases that are of importance on the African continent, and in Ghana, as we've seen in recent times, but we restrict ourselves to these 3, and then we will mention the others as we go along. Again, my name is Dougbeh Nyan. I am a Liberian. I reside in the United States, and I work both in the United States and on the African continent. So my links here are not disconnected. For most times now, over the centuries, Africa has seen a lot of infectious diseases, both emerging and re-emerging diseases. And the continent has also done quite a lot in withstanding and attending to the health burden that these infections place on the human population. We have had Lassa fever outbreaks over the years. We have had typhoid fever outbreak over the years. We have had many others, including yellow fever the diarrhea diseases, etc. In the last almost 2 decades, and prior to that, one of the major infectious diseases that broke out on the African continent was Ebola in 1976 in Uganda, in Zaire, along the Ebola River, for which the disease was then named Ebola virus disease. That disease killed thousands of people, and it was not known what the cause was. Later on, it was then identified and discovered by an African, a Zairean. That information was kept quiet by the medical research community until just 3 to 5 years ago, when he was credited for discovering the Ebola virus. All along, it was being attributed to Westerners having discovered such a virus. He discovered it and identified it. And it is only lately that he's been given the credit. It's unfortunate I don't have his particular name here, but when you search on that, you will definitely find that. Some Google searches are very good with us these days. The virus is very contagious, it's lethal, and it causes what we call hemorrhagic fever. Fever is quite common in our region with many other infectious diseases, commonly for malaria, for typhoid fever, and it causes breakdown or halting of the internal organs and bleeding of the internal organs, as a result of which an infected patient meets their fatality. The virus is a negative strand RNA virus. It belongs to the Flaviviridae family, as is the Marburg virus. And I highlighted Marburg in red because in recent times we've had an outbreak of Marburg in Ghana. It was immediately arrested, and fortunately it did not become widespread. And we'll get into that. There are 5 types of or 5 subtypes of the Ebola virus based on location. The Bujubuju, the Zaire, Sudan, the Reston. There was a species of the Ebola virus found in a place or city called Reston in the United States, and so it was named after that. And then the Taif forest in the Ivory Coast. So these are the 5 subtypes. The case fatality for Ebola is 25 to 90%. 25 at the lower end if the virus is caught in time, the infection is caught in time, and medical attention is given. 90% fatality rate because of the deadly contagious nature and virulence of the virus. The virus is a zoonotic one, and it goes through a zoonotic transmission. It is been documented that the virus resides in bats, and it came from the bat colonies to non-human primates, from non-human primates then to humans. And that's the transmission chain. We will hope not to go into too much of pathogenesis, biochemistry, because there are policy makers who are also watching us online so that we don't bore them. I know when we get back to the laboratory, we shall handle those things. And at an appropriate time, we shall then have a strictly biomedical scientific lecture. And then we can go into the genetics and other aspects. So Ebola, then after the 1976 outbreak, there were a series of outbreaks on the African continent, in Central Africa, in South Africa, and then, boom, in 2013, it hit West Africa. In 2013, in December, in Conakry, Guinea, in a place called Guatemala, there was a young boy who was diagnosed of fever and other maladies that could not be determined. And later on, it was determined that he was infected with Ebola virus. When that was diagnosed, of course, given the challenges the African public health and healthcare system face, the virus and that infection, that in this case was not arrested immediately. And so you had a situation whereby the virus began to transmit from person to person, which was then the human transmission. So it is documented that it came from a bat to this young boy, and from this boy to the members of the family, I think, or one of the parents also fell victim to the virus, and the virus began to spread. Looking at the triangular relationship between Guinea, Liberia, and Sierra Leone, which are the Mana River Union countries, where borders are very fleeting and people go across borders very commonly, you had this cross-border transmission. And so the Mana River Union basin became infected with Ebola, and the war got on its knees when it was described as a public health emergency. As you can see, this particular slide shows cave bats from where either it is directly transmitted to human, or you have these fruit bats where you have the monkeys also, these chimps and monkeys, also living along among these fruit bats, and it's transmitted to the monkeys. And so you have a transmission from the monkeys to cattle and domestic animals, and then to humans. Also, you have bats that are living within the huts in some villages and in some homes, and it is believed that this virus can also be transmitted from them here to the human. And that is the mode of transmission. So you have this chain of zoonotic transmission. Now, during that outbreak, you have a total of 28,000 plus fatal cases, and the deaths were about 11 to 12,000, a combined 11 to 12,000 in Guinea, Liberia, and Sierra Leone. There were other deaths outside of Liberia, Guinea, and Sierra Leone, because the virus then, of course, with the human transport, went also to the United States and Spain and other countries, where then we recorded 36 cases and 15 deaths. The affected countries lacked effective public health response at the time. And this became a very important and critical problem. Why is it that after 1976 and in between 76 to 2013, there was not any attention given to the public health response to the Ebola virus disease on the African continent? Usually when you have Lassa fever, typhoid, we see that the infection is pretty epidemic. But this is one of the largest epidemics we ever had on the continent that took away such a life. And so that question remains, why is it that there was not efforts made in order to put in place mechanisms in public health and other mechanisms that would arrest the Ebola virus and other diseases for that matter? You will note that in 2005, when I lectured for a semester in microbiology, medical microbiology at the University of Liberia Medical School, I then sat with the incoming president at the time and put forth a plan for the reactivation of the Liberia Biomedical Research Institute, like how you have on Noguchi. LIBR, as we call it in Liberia, was cardinally responsible for the production of the hepatitis B vaccine, because we were at the time in collaboration with the New York Blood Bank, where most of the research were taking place, And part of the research was also taking place in Liberia with the chimps and other biomedical bench work was going on. But that research institute became dormant, as you know, during the civil war in Liberia, which took 14 plus years. I describe that as uncivil because of the brutal manner in which it was. I would describe wars as civil when it is based on ideas and good ideas that promote progress in our countries and on our continent. And so I have indicated that we need to put in place public health response mechanism. Because at a time, I had worked and was trained at the National Institute of Health in the United States, worked at various medical research institutes, including the Naval Medical Research Institute, and had this experience to transfer back home. But that prediction that I made, that in the future we may be confronted with an epidemic that may take a lot of lives if we are not prepared. And several years later, in 2014, we had a situation. And the colleagues then began to reflect on those proposals that were made and why they were not followed. But it is not only Liberia, you find that in all of our countries. So, because of that lack of response or effective response and the chaos that was existing, there was global efforts mobilized in order to fight the Ebola virus disease and that epidemic from the US, Britain, Cuba, France, China, Russia, the African Union itself, ECOWAS, and the Mana River Union. During that time, I did testify before the US Congress and called for the establishment of the Africa Centers for Disease Control. We pushed that from our recommendations. After the testimony, we met with senators and representatives in the United States, and the president of Liberia then, Madam Ellen Johnson Sirleaf, was very efficient in collaborating with us in the diaspora. And with other organizations, we finally had the Africa Center for Disease Control established. The disease also went into Nigeria, as you will note. Nigeria quickly arrested it because Nigeria has been confronted with a lot of diseases, if you will note, Lassa fever particularly. So the case that went into Nigeria was arrested. Mind you, Nigeria has a CDC, the Nigerian Center for Disease Control, just as you have the US Centers for Disease Control. But the Nigeria Center for Disease Control was able to arrest this case while the US Center for Disease Control was struggling with the Ebola cases that were in the United States. And the entire country pretty much shut down. So it's only meant for me to indicate to you the amount of potential that we have on the African continent that we sometimes overlook. And so when we proposed the establishment of the African Centers for Disease Control, my intention was, and my purpose was for it to be established and seated in Nigeria, because Nigeria already had a good response mechanism, just as the US CDC. But politics went into it, and of course, it went to Addis Ababa. And the story you can tell for yourself. So you will see that we've been struggling with a lot of pandemics. So after this Ebola epidemic, the next pandemic or outbreak that we are confronted with, or we became confronted with, was this COVID-19. COVID, as a virus, coronavirus, has been around for ages. It's been living with us for centuries. And it has been so dormant. Even on the African continent, we have been confronted with coronaviruses that we may not have taken note of. The common cold we get, the influenza, they're all coronaviruses. They are all of that family Coronaviridie. So for ages, the world has been confronted with coronaviruses and with a lot of other epidemics. And in around 15, 20 years ago, you will note that there was a coronavirus outbreak in Asia. That particular outbreak was transmitted to the United States and Britain. That was termed coronavirus 1. When this other coronavirus broke out from China in Wuhan, the sequence of that coronavirus was compared, or that particular pathogen was compared to the coronavirus 1, and it was found to be about 80 to 85% identical. And so you have this novel virus then designated as SARS-CoV-2, which is the severe acute respiratory syndrome coronavirus 2. Coronavirus 2. This was designated as COVID-19 as the disease by the WHO later on. And it had various variants, including the Alpha, Beta, Gamma, Delta, and the Omicron with which we are currently confronted. Omicron has had BA1, BA2, now we have the BA5, which is much transmissible and deadlier than the previous variants. This is a cartoon of the coronavirus itself with the spike and the spike proteins, the nucleus with the RNA. This is a positive sense single-stranded RNA virus, and it is similar to SARS-CoV-1, and of course, it is similar to the Middle Eastern Respiratory Syndrome coronavirus. They are all of the same family. Globally, we have seen now 606 million cases with 6.5 million deaths to date, according to the data that I saw yesterday from the Johns Hopkins University. So the spike proteins are very important because of their contribution to us developing diagnostics and vaccines and other therapeutics. Kindly take note of that. This is a comparison of the phylogenetic relationship of SARS-CoV-2, SARS-CoV-1, and MERS. These are all of the same family, and you can see their structural identity and their structural design. They have the same open reading frame, 1A, 1B. They have the same non-structural proteins, but slightly reorganized. All three have the 3' region, untranslated region, and the 5' non-translated region as well. As indicated earlier, these are the sequence identities, like I said, 80 to 85%, and so you have only 15 to 20% difference. This is very important in terms of diagnostic specificity and diagnostic sensitivity. It's also very important when you're developing vaccines or other forms of therapeutic intervention, so as to know whether there is cross-reactivity, which will then help whether in a positive direction or in a negative direction. If there is a good cross-reactivity, you are assured that vaccine developed can be used also for COVID-1 and MERS as well. So these are all of very important medical, diagnostic, and therapeutic importance. This is the pathogenesis. Like I said, you can go through that in your literature. But the important thing in this pathogenic cascade is that with the attachment of the spike proteins to what we call the ACE receptors, you have the virus entering the cell, going through all of its biomechanical mechanism, gene translation, and all of that, repackaging in vesicles, and then through exocytosis, pushed out and become available to the infected person. And when you cough out or sneeze, anyone near you who is not farther distance away from you, we say 6 feet distance. I always say 7 feet, because by the time you calculate in your head whether I'm 6 feet away from this person, you would have been farther from them. So if you are not 7 feet away from that person, you could then inhale that and become infected, and then it goes through that cycle. And that's how the infection is transmitted from person to person, from community to community. Now, important here is that when you compare, after we had then done this diagnosis, as you know, there were lack of testing kits, there lack of vaccinations, shortages of vaccination. The United States government and EU government put billions of dollars into their biomedical and bioengineering entities to produce vaccine and to produce testing kits. What did we do on the African continent? We took money, borrowed money from the IMF and World Bank, and it is very questionable as to where the money went to this date. So you will see that we remain unvaccinated as a population in Africa. You look at the United States, and this data is from like July, May. It's an old data, but it still reflects what happens today if you extrapolate. You have 328 million people in the United States. On the African continent, you have 1.3 billion people. You have, in Canada, 37 million. Look at the number of fully vaccinated, 64%, 85%, 84%. In a population of 1.3 billion, you have 8% vaccinated. So what happened to the money that was sourced out to get vaccines and to produce testing kits? These are questions when we talk about pandemic preparedness and response that come into play, because the governments that we are begging all around the world, the US government, the EU governments, to give us vaccines, and then we have these vaccines through Gavi, this vaccine pool, from where we wait for philanthropics like Jack Ma to give us testing kits and to buy vaccines and bring them to us. And yet we get money to produce the vaccines or buy it, and yet we can't buy it and we can't account for the money. So it's a major question for our policymakers who are watching online to look at this data and see the discrepancy in our response and in our effort to fight back this pandemic as it relates to how we were delinquent in fighting back previous pandemics like the Ebola, the Lassa fever, and many other epidemics that have broken out on the continent. Here is where we have certain issues. One, generally, the African healthcare and public health system, we are very weak in national health policies implementation and our preparedness. We have a fragile health system. We lack infrastructure. We have less trained in terms of the capacity personnel. And of course, during these periods, we have this barrier to testing and shortages of testing kits and personal protective equipment that I mentioned earlier. That makes our entire population to be potentially vulnerable all the time. And because we go to church every Sunday and go watch night and fast and pray, we always leave everything to God. Now, it is not that we have not done anything. Certainly, we have done quite a lot, gaining our own experiences. We have had numerous experiences with epidemics. I mean, the African continent should be the master of response to epidemics because we have had the bulk of the epidemics that have taken place and outbreaks in the world. And we have a youthful population that is pretty much over 50% on the African continent. The lockdown did help us a little bit during regulations, and we did improvise with face masks. And guess what? When there was the outbreak, there was shortage of PPEs, personal protective gear, particularly face masks all over the world. The United States, the European countries were also short of testing kits and personal protective equipment. Now, because of that, there was a regulation that these surgical masks that we are using now should only be used by the responders. And for those of us from the African continent, we as Africans, we improvise all the time. So in most of our discussions at the time, because I'm still head of the diaspora COVID-19 focus group, we said, my proposal specifically was that we can make, our African lappas that is sell, we can make face masks out of that. And this was something that they saw was crazy. For every time you are innovative in thinking, what is not the norm and the normal becomes seen as crazy. So you becomes a madman. So I was classified more or less in the brains of my colleagues as that kind of person. Say, why would you think so? It's not made of this material, it is very porous, it is this. I said, something is better than nothing. Because if you have the cloth face mask, we can improvise, use our biomedical engineer, and see what materials can be put together along with that, and it could help in the face of this pandemic. We started producing the face masks on the African continent when we pushed it. The Africa Bio, which I spoke to you about when we organized the conference here in Ghana last year and the year before, we got together and in Ghana, in Liberia, and almost all of the other African countries, but it started from West Africa. Liberia particularly, we started producing the face masks, making the N95 forms to mimic the N95 also, and it did help. Now what happened? Once we started doing that, boom, even the designers like Louis Vuitton and all these people, they started making cloth masks also, but it was not stopped. So you see how the African continent has been innovative, Africans have been innovative, Africans have been very, very proficient in what we do to help ourselves. But when we put forth these suggestions, they are quickly trashed. And my advice to you in this respect, for you, our younger colleagues, is to be confident in what you think. Science is very dynamic. Science is not static. Science is not a Bible. Science is not a dogma. Think and put your thought into practice. As we say, practice makes perfect. So remember that. So my senior colleagues, allow the younger colleagues to think. Let's do that. Let's allow them to think. No, let them bring out all the craziest ideas they can think about. Let them bring it out. Let's try them. I am an example of that experience, as a result of which my own invention came. My first invention was the development of a diagnostic test for ulcers. Now, ulcers, to be properly diagnosed, you have to use endoscopy, and then you go through the laboratory and you do the smear and all that. It is diagnosed through many other means which we will not go into. But my idea when I was at the National Institute of Health in the United States, being trained as a scientist, then after graduating from medical school and going through medicine, became bored with clinical medicine and wanted to get something tested, was that if this bacteria is gastrointestinal, and we can diagnose cholera, Vibrio cholera, through the stool, then there must be a way that we can diagnose this thing through the stool. It was never done. And when I put that forth in a seminar like this, again, everybody thought I was crazy. But my mentor, Dr. Coleman, may his soul rest in peace, we went back after my presentation, and I'm telling you, these presentations at the National Institute of Health, these are Nobel Prize winners, all the award winners you're facing. And so, and I said, Dr. Coleman, if you can increase my budget for research, I could try this thing. Let's just try. And he thought for a long while. He said, okay, let's go ahead. We'll increase the budget. And that's why I'm saying we as senior colleagues and my senior colleagues, we should allow our younger colleagues to try their crazy ideas. You can never tell what comes out of that. And so that's how the improvised face masks also help our continent. in regard to the UFO population, in regards to the number of times we've been infected with a lot of other coronaviruses. And the studies are still ongoing, and we are encouraging our governments to provide funding so that we do these studies to nail down specifically why is it that the African continent was spared. There's something scientific. And I said to them in most of my presentation, the West will not do that for you. You have to do it yourself. The research they are doing is in their own interest. Except because we are all humans, so it applies to us. But specifically, even when drugs are being developed, they have to be tested. If they are not tested on our population, in our genetic pool, they could not work with us, but they could work with them. And that is why you see now companies like Merck, Johnson & Johnson, and Moderna, they have to test those vaccines also in Africa. And we'll come to that because during that time, the French scientists said, let's just take the vaccine and just test it on those African people. Such a condescending statement, which we saw to be racist. I reacted to that. I said that the vaccine should be tested in a multi-center trial that will include nationalities, countries from all over the world, so that we have a good data from that. But not just seeing Africa as a testing and dumping ground and you just want to test vaccines here. We welcome it, but it must be controlled and it must be ethical and meet internationally approved standards. And the WHO Director General Gabriel Yesu also commented on that. Fast forward, our responses have been saddled with political interferences as well. It didn't just happen in the United States with my mentor, Dr. Fauci, who got a lot of beating, psychological beating from Trump. And, this unscientific promotion of chloroquine. We use chloroquine here for years and ages for treatment of malaria. And there was this line of talk that said that chloroquine can cure coronavirus. But, the data showed otherwise. Now, on our continent specifically, as you can see, I highlighted this in red, there was this promotion of this concoction, this COVID organics from Madagascar, if you've heard about it. Yeah. This thing was never tested. Yes, I know we borrow herbs, from our grandfather's days. We borrow the herbs, we take them and all of that. We don't even know the dosage. We don't know what's going in us, how our liver metabolizes that, the toxicity, nothing, no safety requirements. We drink them. So they made this concoction. There was no testing, nothing. And it was being promoted by the president. Then they began to deny that COVID-19 even exists. May so rest in peace, Comrade Magufuli, John Magufuli, who in Tanzania completely denied that that thing doesn't exist. In fact, we tested banana and we saw COVID in banana also. Yes. So any testing kit they bring here, it will not work because the testing kit is faulty. We are not saying that all the testing kits are bad. We know we have to put them through, very rigorous testing and all of that. Then we have displacement and misplacement of public health expertise, particularly like in the United States and in Liberia, my country, where you have people who have practically no public health knowledge are put in place of pandemic, are put in charge of pandemic response. Can you imagine that? And the people who have the public health knowledge are taken out for political reasons. We're not doing politics here for today. Then they also begin to undermine the science and discrediting scientific experts, only to perpetuate their own political policies and aims that don't even relate to the pandemic. Fast forward, there were efforts on the African continent. As I said, right here in Ghana, there's a colleague of ours who organizes with us also. You probably may have heard about him or read about him. He is the CEO of Incas Diagnostics. Incas Diagnostics developed what we call this COVID-19 rapid test. And it is very, very sensitive. The sensitivity of this test when trial was up to about 85% sensitive. I mean, this is an antibody test, rapid test. And from my end, we also have this molecular rapid test by my company, Shopflex Biomed, my startup, which detects many pathogens in a short time, and we will get to that slide later. So we detect COVID, we detect Ebola, we detect Marburg virus. So you have a situation where if you have malaria in an endemic region, typhoid, COVID-19, Ebola, all of them present with fever, right? They present with headache. They present similar symptoms. So when you come to me as a medical doctor, I'm professionally guessing what that could be before I begin to treat. And so you need a test that can specifically detect what is in there, because a patient could be co-infected with one or two. So instead of sending the patient back and forth for 3, 4, 5 different tests, the test that I invented can do all of that and just tell you specifically what that patient has, so you know exactly how to go in instead of going in blindly in treating. Now, the interesting thing in our response, and I brought this slide up to highlight the situation in Ghana, is that about $30 million or so was appropriated for importing rapid tests from, I think, Brussels or one of the European countries by the Ghanaian government. At the same time, INCAS had this test developed. Why is it that the US government pumped in billion, and I'm saying billion, B-R-L-L-I-O-N, billions of dollars? They just give it to these companies. They say, you know what? We know you have your money, but we gave you billions of dollars, go and develop tests. We gave you another billions, go and develop vaccines. That was it. They said, we gave you time, go and bring that back. So Moderna, Pfizer, AstraZeneca, all these companies, the EU government did the same thing. Why is it that our government here in Ghana, I'm a Ghanaian as well, by the way. Why is it that our government did not give at least 10 million out of that 30 million, even 5 million to produce this test, and then we market it even right in West Africa? We are a whole big market on our own. Sometimes we just don't know how we've been treated by the West. We are their market, we are their dumping ground. Two things. And why couldn't we do that? Now, the Ghana FDA, which is very good and one of the best food and drug administrations on the continent, refused to grant emergency use authorization. You remember all these testing kits that we are using all around the world, all the vaccines we have been taking, it's only recently that they approved them, but when they were being used, they were only given emergency use because of the pandemic. So on this emergency, why is it that the Ghanaian FDA refused to grant INCAS diagnostic and emergency use authorization for this? Now, the United States is the world premier country in all of these things we are talking about. Diagnostics, biomedical research, biomedical engineering, along with Europe. This is the iHealth COVID-19 antigen rapid test. If you look here, and I will share these slides with you later, you see the FDA emergency use authorization. You see the check mark here, right? They granted this test. Here. Emergency use granted. I put it in green so that you will actually know that green light is green. Go. They granted an emergency use authorization while we refuse our own product. You know, and this is where when we talk about why is it our response is like that and how prepared are we to meet the challenges of outbreak, it's a question that we will continue to discuss, and for which you, our younger colleagues who are coming up, our senior professors, my mentors who are above me, professors who are here at my little level, we need to do the best we can to continue to talk to our policymakers, to advocate. You know, when you use the word advocate, the first thing they think politically. But nothing you can do other than talking for yourself. You know, we need to advocate. So that's where we had an issue. Now, as I indicated, there were collaborative efforts, you know, in vaccine production. You saw AstraZeneca and Oxford, they tied in with the Korean and Indian production factories to produce their vaccine. Johnson & Johnson was working with Merck. Then there's a whole issue of technology transfer and licensing, waiving of IP. As you will note, if you probably might have read also, there is this thing where African countries or the world will say, Moderna, Pfizer, and all these companies should give their licenses so that the vaccine can be produced in other countries, in Africa. Even if we were to get a license, where can we produce it? You know, these are the questions. You have Cuba and Iran collaborating on vaccine production. But all we were doing was relying on Gavi to mobilize vaccine for disadvantaged countries. It is only now, after all of these efforts, that you have Moderna say is going to start production in Africa. So if you probably might have read recently, one of those plants will be in Ghana here, South Africa, and Rwanda. So that those three countries will be the hub where the whole vaccine production. The question remains, is it just filling the bottles and we say it's produced here? Or are we going into the actual science? And I can assure you that on the African continent, we have astute scientists like your mentors, your professors. The only problem is the resources. Look, in the labs we are in the United States, you can use one box of gloves in 30 minutes. It doesn't matter for them. I mean, we just take the glove. When we are done, we throw it in the trash. We go for another pair. One whole box. So how can a scientist here work if they will have to be cautious of how the resources are utilized? They are thinking about that instead of thinking on whether, you know, instead of thinking on the genetics. How? But in my kind of country, a policy maker will, you know, dip his hand in the coffers and go for vacation in Paris and get married there. You know, and a professor is struggling. The students are struggling. You know, so these are policy issues that we really need to look at when we look at this whole thing. So our argument during these past conferences was that these conferences were held here in Accra for the last 2 years, the Africa Bio, and we really hope that Noguchi can participate in that, where medical laboratories, research scientists have all come together in the last 7 years, and we've been discussing these issues, the science, the medicine, the biomedical research on the African continent. And it's been going very well. The next conference, Celebrate Lab, will be in Abuja, and we will definitely inform you about that. So we have the technical expertise to do these things. You saw Incas producing diagnostic kits. I am from Liberia, produce diagnostic kit that can be, and I tell you something, what I invented was also given to the Liberian government and to ECOWAS. Ghana is part of ECOWAS, right? I gave it to ECOWAS. I said, ECOWAS can come, let us develop this and use it right here in Africa. And so I get money. Since 2016, when I was national orator, they've never come to say, where is that patent that you say you want to give us? Instead, some governments started to undermine. But the Rwandans, the Egyptians, and other African countries showed interest to the extent that Rwanda provided me a very huge laboratory space, 2 times this small. So anytime you get funding, come here and do your work here. You know, so that's the only country that has been very generous so far. All during this time of the COVID-19, we kind of neglected that we had malaria, we had typhoid, we had Lassa. Then also there was a brief outbreak of Ebola in Guinea, you know. But what happened? From the time we developed the CDC, right, and we put in place, we also talked about creating public health institutes in the regions and in the countries. So they were able to immediately arrest it, so that it didn't become an issue. You see how when you think far ahead, and that's how these countries that we are running behind, that's how they think. They only plan ahead of themselves. They don't wait till things happen before they react. So all that time, something else was happening. Shortly before the before the coronavirus outbreak, there was an outbreak of human monkeypox virus in Nigeria. And there were sporadic outbreaks also in Central Africa. Nigeria started to deal with it simultaneously as it was handling the coronavirus response. Now, monkeypox, as you will note, is definitely a zoonotic smallpox-like virus, just like smallpox. It causes what, it's caused by the monkeypox virus, which are all of the genus Pox virus. It's a double-stranded DNA virus. The previous viruses we looked at were RNA viruses. This is a DNA virus, just like the hepatitis B virus, which is also DNA virus. And it has about 1.6 kilobases of genes, or you can say 196,000 base pairs. Again, this orthopox virus genus also comprises variola virus, where this smallpox is also categorized, vaccinia virus, and cowpox. Now, they first recognized this virus in 1970, where again, in the Democratic Republic of Congo, the same, which was Zaire in those days. 1970, since that time, 6 years after that, when we had an Ebola outbreak in the same Zaire. And all that time, why is it since 1970 to 2018, when there was an outbreak in Nigeria, we did not have a vaccine produced on the African continent, but we had smallpox vaccine produced in various countries, which they sent to us, and we vaccinated, and the WHO declared smallpox, eradicated, right? Just as smallpox was being eradicated or declared eradicated, you have monkeypox showing up. And these viruses share similar genetic sequences. So they borrow from each other, just like we borrow, soup to eat and food, from each other. They do the same thing, and that's where we have to look at the similarities, where I spoke about the similarities in those coronaviruses earlier. So why I said that we didn't develop a vaccine that time? You have had several outbreaks since that time, in both West Africa and Central Africa. And like I said, in September 2017, 2018, there was the re-emergence of this virus in Nigeria. Today, we have this virus all over this country. All over the world. If you see these areas, these are the areas where the monkeypox virus outbreak was pronounced. And here, these are all smallpox viruses. So you can see that it is certainly global. Now, during that time, you had most cases now being reported in Europe and America and many other countries in the world. And these cases are due to have been reported in people that didn't even travel to Africa. So this is the interesting thing. These didn't travel to Africa, yet they contracted this disease. So you can see the global nature of it. And most reported cases, interestingly, involve mainly, but not exclusively, men who have sex with men. And this is where you have another public health problem. And for our kind of countries and our culture, this is where you have a cultural problem. So this is the monkeypox virus. This is what it is. Up to now, we have 19,000 cases with 76 in 76 countries. It's also preventable with the smallpox vaccine. Now, this is a test that I developed, which detects SARS-CoV-1, 2, MERS, Ebola, and all of that. The test results can be given in 10 to 40 minutes, easy to use, very accurate, and it has an accuracy, sensitivity of 97%, specificity of 100% with the cohort tested alongside this FDA-approved test as a comparison with these numbers. It's been validated and published in 3 major scientific newspapers, international news magazines, in Nature, the International Journal of Infectious Diseases, and Clinical Infectious Diseases. It's been granted 2 US patents. This was the first patent, which is the main patent, the multiplex real-time isothermal detection and identification of bacterial, viral, protozoan, nucleic acid. So it covers quite a lot. And you have the second patent, which was recently issued in April 2022. This one was issued 4 years ago, and this was issued just a couple of months ago. And this also has to do with methods for rapid detection and identification of viral nucleic acid. So, what are our problems that we can summarize? One, the lack of funding, support for research and development in our countries. Lack of infrastructure, like we were saying earlier, research materials and tools, limited or conditioned research network. If you have to collaborate with somebody, if you want funding from Europe, they say you have to collaborate with somebody in Europe before we give you our money. So if you don't have anyone, your idea stays in you. These are limitations in networking. There's a limited or lack of opportunities for our younger researchers. Then we have our senior professors have this heavy academic instructional load that they may not even think more about research, they're thinking about the academic side. Then, of course, there's a lack of interest in research as a whole with the policymakers. They just don't know what you're talking about, and they don't care. So, here is it. Here are some facts about Africa that we have to look at as we talk all of this. Africa has 1.2 billion people, 14% of the world's population. And that age is between 18 to 30 years old. We have our population is almost like half youthful. Then the economic growth is projected to be 4%. The GDP 2.6 trillion. When you look at all this data, why is it then we can do all of these to tap into our own GDP, and we'll have people tapping into our GDP? So it makes a very big problem. While we were borrowing all of this money, our same GDP was going down while we were heavily in debt. So you see the disparity for which we are strangulated. So they extend that if a researcher goes to ask for money, they don't even want to know why you want money and all of that. I would therefore recommend, and you may add to these recommendations, we need to do training of public health responders. That are described as the Africa Medical Response Corps. During the time I won the award here in Ghana, that African Innovation Award in 2017, where Philippa and I won the awards, President Akufo-Addo was just right next to me, and I had a chance of chatting with him, with the Honorable President, and I whispered some stuff in his ears and said, look, we need to produce things here, and please, can you increase the GDP for research? To his credit, and I will send you the article, to his credit, he went back and increased the GDP by 2 more percent. It was 1%, and he increased it by 2%, it was published. So, so you see how on the stage chat, I did not go to his office, just right on the stage. So, and we need to develop, we need to provide infrastructures and logistics. We need to develop robust testing capabilities in Africa. Because when we talk about responding, we need to develop those things here. INCAS is right here in Ghana. We need to encourage indigenous technology transfer. You know, like the technology we are in America, we as Africans, we develop them. Let's encourage, because when we are there, we think about what's happening here. That's how we think. So we should support collaborative research. There must be increased funding for biomedical engineering, biomedical research and development. We should support local innovation and local manufacturing so that the economy is exporting and not just importing. And that would be medical devices. I mean, they can make other things, but let them make medical devices and vaccines and medicines. Our community vaccine programs for Ebola, smallpox, and monkeypox, since we are in that pandemic now, should be enhanced. And so, if all of this is done, I think we will be getting somewhere. Ebola virus, and particularly this coronavirus, is not yet over. I would still encourage you to get vaccinated. Continue to wear your mask. Maintain your physical distance as much as possible. If it's possible, when you go to the Accra market, if you can maintain that physical distance, good. Keep washing your hands, and you know, we will continue to combat this COVID. This is my team. This is Kalilu, who is here with you. He's on our team. He studied in the U.S. as well, and we've been finding a place for him, so we want to thank the University of Ghana, through which this program is here, and he's here doing his PhD in infectious diseases diagnostics. This is our team, and so look at the kind of laboratory that I work in. You know, now when I come here, when I say here, I'm not talking about Ghana specifically. If you don't have, look how these shelves are packed up. Sometimes we have nowhere to put new stuff that we bring, you know. So, but if you don't have this, how do you work? It's a problem. I want to thank all the places that I have gotten my training, my education, the University of Liberia, the NIH, U.S. Food and Drug Administration, the Humboldt University where I studied medicine, University of Pennsylvania where I also work, and the African Innovation Foundation that have been able to promote our work. I really want to thank you for this opportunity to speak to you.
Thank you very much, Dr. Nyan. And as you can see, he's not crazy. He just has crazy ideas, and a lot of them. Because of time, let me just give the opportunity if there are any burning questions. If anyone has any questions, let's have them. Let's make it very short, straight to the point, no preambles.
When they developed the vaccines, they ordered all of them to make sure that the vaccine will go about 10 to 100 times the weight. If they among themselves, they were stealing from themselves. They were expecting us to fall on the ground dead. And they refused to give them the class. Then when there was an overload of vaccines, they wanted to dump them on us. The number of people that died, that is vaccinated is 8%. Why should I get vaccinated? If after all this, I didn't die. Why should I bother myself?
Let's take 3 questions.
Thank you. So since we are in Africa and money is very important, how much does it cost for your test to run it in the hospital? And number 2, what is the setting? Can I do it on my desk or have to get a lab to do it? Thank you.
Did you get that?
Yes, I got it.
Good. Do you have a third one? All right.
Thank you for the presentation. I just want to find out whether the reagent is used in a closed system. It's a closed system operation reagent or open system.
Okay. To the first question, To the first question, certainly we saw the misapplication of the vaccines on the African continent. In fact, some of those vaccines expired. The act of anyone asking, why should I even get vaccinated when not many people die and we only saw 8% vaccinated and I'm still alive? Everybody go out in the street, but you don't ever know when there will be an accident and a car will hit you down. So you always have to be what? Watchful. You always have to watch the traffic light before you cross. All of those things. I'm just giving a simple analogy. Our immune systems, we don't ever know when our immunity will give way to a strain that we may come across that will be even far deadlier. We saw what happened in South Africa. Because it didn't happen probably in Ghana, Liberia, and Sierra Leone, does not mean that it could not have happened here. We saw what happened with Ebola in Liberia, Sierra Leone. So it has to do with our immunity, that we don't know at what point our immunity may give way to a more virulent strain. And that's why vaccination as a means of prevention is always important. To the second question, if you can just recapitulate that, I will appreciate it. The cost of the test.
And where can we do it?
Yes, you can do it on the bench top. You can do it in the lab. It's also mobile. You can use that in mobile vehicles. In fact, the test is made such that when you collect samples from one village and go into the other village, you can do that in a car as you go to the other village to collect the next sample. So by the time you get there, the results are out and you can send it out to Central. So it's very versatile, the question that you asked. So you can use it in an open system, you can use it in a closed system. And that answers the last question. The cost estimate is such that we target servicing what we call poorer countries, so to speak. And so whereby one test of HIV, for example, may cost you $125 to $175. The test that we develop that can test the 3 at the same time may cost you like $25. So if you're getting 3 results for $15 to $25 as opposed to one test for $175 to $200, look at your pocket and see which benefits you.
Do you have any others?
Yeah.
Can you project?
Yes, I can. So thank you very much for your presentation. I wanted to find out, so for these 3 or 4 tests targeting different viruses, what samples, because for the various viruses you may need to use different samples. So in that case, how do you combine samples in this test kit?
That is correct. The extraction of the nucleic material is what is important. For Ebola, you need blood samples or saliva samples. For malaria, you need blood samples, right? For COVID-19, you need nasal swab samples or oropharyngeal swab samples. So it is the extraction process that is slightly different, but all of those extractions can go into the one test, and that one test can determine what is in there and what is not in there.
So if I get you correctly, you have to do your extraction first.
Yes.
And then you combine the nucleic acids in the test.
You can put all the nucleic acid in that one test. Equally so, we developed the test in a way that even for the saliva sample, you can use it directly in the test without even nucleic acid extraction.
Yes, if that is for one specific virus, fine.
Yeah.
Where you are combining, you
Right.
still will have to do the extraction.
The extraction, yeah, which makes it more sensitive. Because you know, the better you extract, and you have the nucleic acid, itself, be it RNA or DNA, the more sensitive your test is, and you can get results.
Okay, any others? So let me just ask, what is your next crazy idea to test, and how can we support that?
We are currently at the stage of the pre-production stage. We're looking for funding in order to go to production immediately and then go to the market. That's the stage at which we are. We are looking at various other pathogens, because we target 23 major pathogens globally. So the test is globally applicable. That includes HIV, hepatitis B, hepatitis C, which are always like co-infection. You have co-infection in those. And then in our region, you have the malaria, typhoid, Lassa, and of course there's Marburg and Ebola. So that's where we are. There are other ideas that are coming up. Those are restricted to some trade secrets that we can't really talk about.
He only tells his friends, those ones. So.
And that is why, in fact, one of my last statements will be that there are a lot of good ideas coming out of Africa. We also have to protect our intellectual properties. It's very important because these ideas are here and people come in and just take them and go. All this pandemic, there were a lot of things you suggested to some people, they just took it and went. And then they produce it there and they say they invented it. During the Ebola outbreak, there were innovative Liberian doctors who developed certain, or treatment protocols. And as soon as you knew, some Westerners were publishing it that they developed those protocols, simply because they were just on the ground and, doing one or two hands-on. So we have to protect our intellectual property.
Yeah. Okay, I think we are all very satisfied. So just join me in thanking Dr. Nyan very much. I think this has been very interesting. And
also thank you to Dr. Black-Blanche, who is his contact here, and brought him to the institute. Thank you also very much. Just to sum up, he made some very important points. And he said young people. Now, I don't know if I qualify for young people or not. But let me say to the young ones, he said, think outside the box. Okay, that is very, very important. Don't limit yourself to what you can do or what you can think. There's always this that I tell my students. There is a scientist who sacked his postdoc from his office because the postdoc said the carbon, when you have an organic compound, okay, the arrangement of bonds around the carbon is tetrahedral. He thought it was planar. He sacked him for thinking that, and 100 years later they found out that was the case. All right? So don't let anyone tell you you are crazy. Just think outside the box. All right? Thank you very much for coming.
Would like to take some pictures.
Oh, okay.
Look, we see, you have to come, we'll have a picture. And then we. Yeah.
Yeah.